Hello students, today's class is on types of immune response part 3. Introduction. Immune system is a defense system which protects animals from invading pathogenic microorganisms and cancer. Once a foreign organism has been recognized, the immune system recruits a variety of cells and molecules that mount an appropriate response called as effector response to eliminate or neutralize the organism. In this way, the system is able to convert the initial recognition event into a variety of effector responses, and each is unique for eliminating a particular type of pathogen. The large molecules, usually proteins, on the surface of cells, viruses, fungi, bacteria, and some non-living substances such as toxins, chemicals, and foreign particles are referred to as antigens. Antibodies are the substance produced by the immune system in response to the presence of an antigen. The major agents of adaptive immunity are lymphocytes and the antibodies and other molecules they produce. Immune response is the immunological response which originates when the immune system is activated by an antigen or the response to pathogenic microorganisms and its products, as well as autoimmunity to self-antigens, allergies, and graft ejections. The immune response can be transferred via serum antibodies introduction from sensitized to unsensitized individuals. Now let's see what is immune response. The immune system controls the immune response. The immune response protects the body from bacteria, virus, parasites, pollution, fungus, etc. It is a complex mechanism and many cells interaction compromise the immune response. The B and T cells work in coordination during an immune response. An immune response is what the immune system does when the body is confronted by an antigen. It is an elaborate interplay between antigen, non-specific defenses, and B and T lymphocytes. Immune response can be humoral or cell-mediated. After an antigen-antibody interaction, certain normal serum proteins known as complement are activated. The first contact with an antigen sensitizes the affected individual and promotes the primary immune response. Next encounter of the sensitized individuals with same antigen results in a more rapid and massive reaction called the secondary immune response. It is most expressed in the level of circulating serum antibodies. In this process, main cells involved are the T cells, B cells of lymphocytes, and macrophages. These cells produce lymphokines that influence the other host cells' activities. B cells mature to produce immunoglobulin or antibodies that react with antigens. At the same time, macrophages are processing the antigens into immunogenic units which stimulate B lymphocytes to differentiate into antibody-secreting plasma cells, stimulating the T cells to realize lymphokines. Now coming to the types of immune response. Immune responses to antigens may be categorized as primary or secondary responses. The primary immune response of the body to antigen occurs on the first occasion it is encountered. Depending on the nature of antigen and the site of entry, this response can take up to 14 days to resolve and leads to the generation of memory cells with a high specificity for the inducing antigen. The humoral response mediated by T cells with the help of T cells produces high affinity and antigen-specific antibodies. The secondary response of both B and T cells is observed following subsequent encounter with the same antigen and is more rapid, leading to the activation of previously generated memory cells. This has some quantitative and qualitative differences from the primary response. In a B cell primary response to a thymus-dependent antigen, the immune system selects B cells with a high affinity and specificity for the antigen, and this becomes memory cells. In a secondary response to the same antigen, memory cells are rapidly activated. This process is quicker and more effective than the primary response. A single injection of antigen helps in sensitizing or priming of immunocomponent cells, producing particular antibody instead of an elaborated high level of antibody. Effective levels of antibody are usually produced by only subsequent injection of antigen. The antibody response to an initial antigenic stimuli differs qualitatively and quantitatively from response to subsequent stimuli with the same antigen. Now coming to antibody production in primary and secondary immune response. The primary immune response functions by making antibodies when an antigen comes in contact to the immune system for the first time. During this time, the immune system has to learn to recognize antigen and how to make antibody against it and eventually produce memory lymphocytes. This figure shows that the first antibody produced is mainly IgM, but small amount of IgG are also produced. The secondary immune response occurs when the person is exposed to the same antigen for the second time or third, fourth time, etc. At this point, immunological memory has been established and the immune system can start making antibodies immediately. So, in secondary immune response, mainly IgG antibody is produced in sufficient amount and sometimes small amount of IgM are produced. Other immunoglobulins such as IgA and in the case of allergy, IgE are produced. This figure shows the differences in the primary and secondary response to injected antigen. It reflects the phenomenon of immunological memory. When an animal is injected with an antigen, it produces a primary serum antibody response of low magnitude and for short duration, peaking at about 10 to 17 days. A second immunization with the same antigen results in a secondary response that is greater in magnitude, peaks in less time, that is 2 to 7 days, and lasts longer, like months to years, than the primary response. And we can compare the secondary response to antigen A with the primary response to antigen B administered to the same mice. Now coming to the involvement of lymphocytes in immune response. This figure shows that memory for a primary response can be transferred by small lymphocytes. Recipients are treated with a dose of X-rays which directly kills its own lymphocytes as they are highly sensitive to radiation but only affects other body cells when they divide. Thus, the recipient will permit the function of the donor cells. The immunologic potential of the transferred cells is expressed in the recipient. Thus, any immune response will be of donor and not recipient origin. Small lymphocytes are taken from an animal given a primary injection of tetanus toxoid and transferred to an irradiated host which is then boosted with the antigen. A rapid, intense production of antibody characteristic of a secondary response is seen, and influenza hemagglutinin is used as a control antigen to exclude the possibility that the first antigen injection might exert a non-specific stimulatory effect on the lymphocytes. Now let's see how is vaccination related to immune response. The infectious antigens like microorganisms are present in our environment, and it is likely that we meet again. The first contact with the antigen clearly imprints some information and imparts some memory, so that the body is prepared to repel any later invasion by that organism and a state of immunity is established. This is the basis for the development of immunity and the reason why we rarely suffer twice from such diseases as measles, mumps, chickenpox, whooping cough and so forth. The same basis is applied in the production of vaccines. This figure shows the basis of vaccination as illustrated by the response to tetanus toxoid. When the tetanus toxoid is given in the form of a vaccine, it leads to primary immune response where small amount of serum antibody is produced, and later exposure to the toxin in the subsequent natural infection boosts the memory cells, producing high levels of neutralizing antibody which are protective. So this is how the mechanism of vaccination works. Now coming to autoimmunity. Autoimmunity is a system of immune responses of an organism against its own healthy cells and tissues. Any disease that results from such an aberrant immune response is termed as autoimmune disease. Few prominent examples are Addison's disease, rheumatoid arthritis, sarcoidosis, Graves' disease, Hashimoto's thyroiditis, etc. So we can say that autoimmunity is the abnormal functioning of the immune response. Now coming to conclusion. In this lesson, we have learned that immune response is controlled by the immune system. Immune response protects the body from microorganisms. It involves varieties of cells and molecules which remove the foreign invaders like B and T cells, macrophages, cytokines, etc. Lymphocytes are the cells which provide memory. Immune response is activated once it is encountered by an antigen. Vaccines are also developed in such a way that they evoke the immune response. Although most immune responses are beneficial, but few can be harmful, as in the case of autoimmunity. Immune response can be divided as primary and secondary immune response. The primary response is slow, sluggish and short-lived with long lag phase and does not persist for long time. The secondary response is prompt, powerful and prolonged with short or negligible lag phase and with higher levels of antibodies. Thank you.